how can we stop liver cancer? Hepatocellular carcinoma, or HCC, is the most common type of liver cancer. It is one of the deadliest cancers worldwide. In our study, published in Gut, we present a novel strategy to harness the immune system by isolating T-cell receptors, or TCRs, that specifically recognize tumor mutations in hepatocellular carcinoma. Hi, I'm Anna Pasetto, a scientist at Karolinska Institute. My research aims to train the immune system to find and kill cancer cells by targeting neoantigens, mutated proteins found only in tumors. Together with my PhD student Panagiotta Maravelia and the clinical team at the Liver Transplantation Unit at the Karolinska University Hospital, we designed a study to see whether T cells from patients with HCC could recognize this tumor-specific antigen.
I'm Panagiotta, and I will show you how we did it. We used a pipeline that combined tumor exome sequencing, computer-based neoantigen prediction, personalized T-cell screenings, and single-cell sequencing to find T-cell receptors that can recognize neoantigens. We collected T-cells from different sources: the tumor itself, blood flowing through the explanted liver, and nearby mesenteric lymph nodes. Why so many sources? We used different T-cell sources because we wanted to think beyond the liver. The liver's environment is known to suppress immune responses, especially when it's the first place the antigen is encountered. Therefore, by looking outside the liver, we hope to find stronger, more reactive T-cells. With this approach, we found several T-cells that could recognize tumor-specific mutations, especially in the liver flash or lymph nodes of multiple patients. Let me walk you through one example, a case where the T-cells targeted a unique mutation in the SBNO2 gene found only in the tumor cells of one patient. This mutation changed the SBNO2 protein in a way that made it recognizable to the immune system. In fact, as seen on the left graph, the T-cells responded only to the mutated version, not the normal one. When we isolated and sequenced the reactive T-cells, we discovered 3 TCRs that were much more abundant in the reactive population than in the non-reactive one. We cloned these TCRs and retested their function, this time by genetically introducing them into fresh T-cells. 2 of the 3 TCRs showed clear, specific recognition of the mutated antigen, confirming both their reactivity and specificity. We could also show that the reactive TCRs expressed unique phenotypic and functional markers, distinguishing them from other bystander T-cells. Overall, these findings show that neoantigen-specific T-cells are not only present in patients with HCC, but can be identified, isolated, and engineered, paving the way for personalized immunotherapies.
So, what happens next? The road to the clinic is still long, but this work paves the way for TCR-engineered T-cell therapies tailored to HCC patients, with a personalized approach grounded in their unique tumor mutations. Thank you for watching, and please find our full article in Gut.